Welcome to our next set of videos. This particular one we're going to look at how to prepare an onion slide to look under a microscope. Onions are particularly useful because you're able to peel off a layer of skin on an onion that is only one cell thick, and it's really easier for us to see the parts of a cell that we talked about in the previous video. So what I'm going to do, first of all, we're going to open up an onion, take off the skin a little bit. Actually, what I'm going to do first of all is cut off the ends, like so. Cut off the second end, like so. And then I'm going to take out a wedge of onion. So we've got the different layers here. Now what is really useful with onions is that on the inside of each layer, if I cut off a little square section, I'm going to make it easy enough, right size to manipulate. Each layer of an onion on the inside, tease it off, sometimes easier just with fingers to be honest, but here we go. If you peel on the inside, you can get one layer of onion off, and that layer of onion is one cell thick, which is going to be really useful for us to see inside, because light can travel quite easily through that one layer. So your stage one, what you've got to do is you've got to cut open your onion cell, you've got to take off a layer, and you've got to put it onto a piece of glass. This piece of glass is called a slide. You flatten it out, make it nice and smooth. Second, if you notice, if you look through this through the camera, you can see it's fairly transparent. You can't really see it. And the parts of the cell that we want to see are really hard to see. So what we want to do on that, to make everything stand out, is to use a stain. Now the stain we're going to use today is iodine, which is very good. Those of you know from other topics, it stains certain things like starch, it will change colour. So if I put this on, because it stains various different parts, I can put a couple of drops on that, put one more, and that will start to stain the parts of the cell that we want. Now we don't want to look at that direct like that, because we don't want the iodine getting onto our lens on the microscope. So what we're going to do is we're going to put one of these extremely thin layers of glass called a cover slip. Very, very thin, very, very delicate. Now, when I put this onto here, I don't know if you can see this. Can I get the camera around, Chris? When I put it on, you can see the iodine gathers at one edge. So once it's there, because it gathers there and you put it down, we let go, you can see it spread all the way throughout the slide. Now, in order to see that, I don't know if you can see that clearly, but inside there, we've still got some air bubbles where there's a clear part. Now, best thing I advise is just get your finger on it, squeeze it around, and the air bubbles soon come out to give you a more consistent yellow. Now, this now is look at, excuse that little dot there, that's one of the onion skin that's there, so we're going to ignore that little dot, but we're going to focus on a patch where we think there's no air bubbles. So there's a little area there that we're going to look at the microscope on. So we take our microscope. Now this is run by battery, so I can turn the light on immediately from below. Now hopefully you've watched the how to use a microscope section, but just a little reminder, we always start on the smallest of the lenses, which gives us a 40 times magnification. We'll find it easier to find the cells with this one first. We're going to put our slide onto the stage here. We're going to put it just over the light. Once that's there, another little hint, always have the stage as high as it'll go. The reason for this is when we're going and we're going to look under it, what we don't want is the slide to smash into the lenses. And if we start at the top and move downwards, that can't happen. So we always start with the slide with the stage at the top. And we put one eye into it, and as we look down, we change the focusing knob until you can see what looks like brick-like structures. Appearing on your screen now is a picture of what you can see. So there will be an image now of some onion cells. You can see all the bits of a cell that you said we should identify from a previous video. You can see the cell wall, the cytoplasm, the membranes, and quite clearly the nucleus. Vacuoles are harder to see, but you won't find any chloroplasts. if you could say the reason for that, that would be brilliant. There is a reason why onion cells do not have chloroplasts. After you've seen this section, there'll be a little question that's going to appear on the video. The question is a typical 4-mark question that you might see in the exam. The kind of question asks, describe, for 4 marks, the stages you would use in preparing an onion slide. Please use this video, write down 4 steps, and what I'll do is at the end of the video I'll include where you get your 4 marks from. Thank you very much for watching.